What's good, y'all, and welcome back to another edition of Mr. Fitzgerald's biology class. Today we're going to dive into Mendelian genetics and kind of review all those components within that unit that was all about Punnett squares and all that fun. So let's go right into it. So real quick, remember there's some vocabulary with this overall. So this would be a Punnett square representing a monohybrid cross because we're looking at one trait, either the dominant or the recessive here. And these would be variations of a gene, also known as an allele. So an allele, usually for us, is represented by a letter, either sometimes capital, sometimes lowercase, sometimes it's two letters, depending on if it's a linked thing like an X-linked gene or something like that. The genotype will be the combination of alleles that something has. So this one has a capital letter and a lowercase letter. This one has two lowercase letters representing its genotype, letting us know kind of what overall DNA features it has. Its phenotype will be what it shows. So for this one, it's going to show as a yellow color, and this one's going to show as a green color. Something that is dominant is one that will show over another. So yellow, as you can see here, is dominant over green because that's the one that is showing in the phenotype here. And green must be what's called recessive, or the one that's hidden unless it's with another recessive trait. So overall, I have two recessive traits here. All right, so that'll bring us to another kind of concept idea within this. It's called the law of segregation. So previously on this last slide, if you notice, we kind of have the genotypes being broken up here. So the alleles are put next to their own boxes. And that's really kind of showcasing this law of segregation, that the alleles actually separate. So when we generate our sperm and egg cells, our gamete cells, during meiosis, we'll actually kind of divide up the two alleles within the genotype to kind of separate them out, and that gives us some genetic variation overall, some variety within our species. Now, what's cool about this is that diploid cells, like a skin cell or an eye cell or something like that, will have two copies of every allele. So they'll have two alleles per trait representing the full genotype, whereas sex cells will only have one allele. So that way, a sperm cell has the one allele, an egg cell will have the other corresponding allele, and then we match those together to make our genotypes. Okay, and that's kind of what's being done with Punnett squares. Now, independent assortment is when we have the big 16-box Punnett squares. And this is the idea that one allele doesn't dictate what the other one does. So for instance, here I have Y's and R's. So when we break those alleles up, what you get for Y, if it's a capital or a lowercase, does not tell you exactly what you get for R. You can have either a capital Y with a capital R or a capital Y with a lowercase r. So really, depending on which combination you get is what the outcome would be. Because the Y is capital doesn't mean the R has to be, and vice versa. Okay, so you get a lot of variety in here, and that's because it's all random. This thing is a random distribution, and this one is a random distribution, and then we pair them up accordingly. Okay, so some other vocab here. Homologous is when the alleles are the same. You either have like two capital letters or two lowercase letters or two of the same letter when you have incomplete or codominance kind of coming into the fray. Also, heterozygous would be when you have mixed letters. So usually one uppercase, one lowercase, or two different letters, something that would show a little bit more variety or mixture. So homozygous are your purebreds or your true breeding things because they have the same alleles within their genotypes. It's always homozygous, or two of the same for dominant, two of the same for recessive, or two of the same allele. And then heterozygous would be any that have a mixture where one's capital, one's lowercase, you have some dominants, some recessives, you have maybe different colors, like you get pink as a blend between red and white, or checkered as a phenotype from black and white. So you get some variety. All right, so real quick, just a very fast practice problem here, looking at a simple monohybrid cross, just to remind you of how a Punnett square works. So if tall is dominant to short, what is the genotypic and phenotypic probabilities of a homozygous recessive plant being crossed with a heterozygous plant? All right, so a homozygous recessive plant would be two lowercase letters, and a heterozygous plant would be one capital and one lowercase. So if we fill this in real quick, let's put our homozygous across the top, our heterozygous on the side. Right? I'm splitting up the alleles, showing that law of segregation here, and then I'm getting basically each one comes into the box that it corresponds with. So the capital T comes all the way across, and the lowercase t's in both of these scenarios come down. Lowercase t's come across, and the lowercase t's come down. Excuse me. All right, so the genotypic ratio that I get here is kind of the letter combination. So I have a heterozygous ratio of 2 out of 4, or 50%. So 50% chance of getting heterozygous. I also have a 50% chance of a homozygous recessive as well. Now that brings us to our phenotypic ratios. Now our phenotypic ratios are going to be what's shown as a result of this. So because I have this trait here that's dominant, this capital letter, it's going to show that dominant trait, which it tells us in the beginning of the question here that tall is dominant to short. So I would expect all 50% of these to be tall, and I would expect these that are 2 recessive traits to be short. All right. So that's how I can get these genotypic and phenotypic ratios. Now in this case, the genotype and the phenotype did come out to be the same ratio, right? 50 and 50. That always doesn't have to be the case, but in this case it just happened to be that way. Another possibility it could be is incomplete dominance, and this is where you get a blending of traits. So if you have red flowers and white flowers, right, showing these different phenotypes here, as well as their different genotypes, and they're representing them as CR and CR, showing that it's the color red and the color red that we're mixing together to get red, and the color white and the color white that we mix together to get white. And when we have pink, we have one color red with one color white allele, and those blend together to make that pink. So this is where one trait is not completely beating out another. They're both being expressed and blended together, so we get a phenotype that's a mixture of the two. Okay? Another one that's kind of a mixture or heterozygous kind of thing is codominance, and this is when both alleles express together, although there isn't any mixing. So you have black chickens in this example and white chickens, making this checkered chicken. Now it's really hard to see, but imagine it kind of just being full-on checkered. The fancy name for that is called permanent. So you can get both traits showing at the same time. Remember that prefix co means together when dominant needs to express. So you're going to see both black and white here, whereas in incomplete dominance, you see the blending of them. So it's kind of a mixture between those, okay? Now, another one that comes up is blood types. Now, blood types is an example of what's called multiple alleles, where instead of having just 2 allele types like black or white or red or white or tall or short or anything like that, where it's kind of like a one or the other, now we have another possibility that comes in. So we can have, A's, we can have O's, and we can have B's. So because we have 3 different alleles it can be, we get more combinations. You could have 2 A's together, like a homozygous A group. You could have a heterozygous for A. You could have a codominant AB situation. You can have a homozygous for blood type B, heterozygous for blood type B, and then homozygous for blood type O. And more often we write the blood types as this, so you might see this on tests or quiz questions. And why this one is important is it shows you which one's dominant and which one's recessive. So in this case, A, the allele for A, and the allele for B are both dominant alleles. So if they're ever paired up with O, which is recessive, they will show. So previously when I was kind of rattling off the different blood types, I would say heterozygous for A here because it's going to show blood type A because A is dominant, O is a recessive trait. Same thing here, heterozygous for blood type B because B is dominant, O is a recessive trait. So this kind of way of looking at it does help you kind of understand which one's the dominant and which one's recessive. So I'll kind of mix those in right now. So when you have A's and B's that are with O's, then those A's and B's are going to show because those are my dominant traits. So allele A's and B's are dominant. So here's the way of showing it, textbook style, where the IA with a little I, that'll give you blood type A, or a lot of students will just write it as A's and O's because it's easier for them. Either way works for me. Just remember that O is the recessive trait, okay? Same thing over here with IB with a little I or BO, either one gives you blood type B, and then OO or little II would be your blood type O. Now, when you get A and B together, that would be a codominant situation. So in there, you would actually have a with a B, right? I had a little mistake there, but you have a capital A with a capital B here, representing that they're both dominants, or you could just simply write them as allele A and allele B, representing phenotype blood type, phenotype AB for blood. All right, let's do a quick blood type one real fast. So if a parent is heterozygous for blood type A here, and homozygous for blood type O is the other parent, what would be the cross? So blood type A's parent right here that's heterozygous, I got the capital I with the A, I got the lowercase I. The blood type O would be 2 lowercase i's. So I mix those together, and overall I got a 50% chance for blood type A and 50% chance of blood type O based on this, right? Because A shows over O, and O is a double recessive, so that's the only way it shows up. All right, now the hard one, dihybrid crossing. Okay, dihybrid crossing is when we're looking at two traits at the same time, and what's the probability of both of them being passed on. Now, one thing that always comes up is they usually ask you questions about either how to figure out what goes on the outside of the Punnett square for these, or what's the outcome in a certain scenario, and this is that certain scenario. So what is the probable offspring of a heterozygous parent for both traits if it's allowed to self-pollinate, okay? So heterozygous means that both traits are going to be one capital and one lowercase. And it's telling here us that dark is dominant to light and tall is dominant to short. So capital, lowercase, capital and lowercase for that dark and also for that tall trait. Okay, so let's kind of look at it for the big 16 Punnett square boxes. I went ahead and color coded these so it's easier to kind of see where I got them for when I do the outsides. Now, the key thing is even though there's 4 letters here and there's 4 blanks here, it's not 1 letter per blank. Because if you did that, you would only end up with 2 letters in each box. Well, it starts with 4, so everything needs to end with 4. So with that being said, we need 2 letters to represent the tops and 2 letters to represent the sides. This will give us 4 in each box at the end. Now, how do we determine that? Well, we need 1 from the D trait and we need 1 from the T trait. And how we do that is usually through a process called FOIL. And you can kind of look at it simply, where if I take the 2 capital letters and I put them together, right, I'm going to get capital D and T. If I take the capital D, since I already started with that one, and match it with the corresponding lowercase t, because that's a possible outcome, I could get a capital D with a lowercase t. Or I could have the lowercase d on the inside pair up with a capital T, or I could finally have the lowercase d and the lowercase t. So this is FOIL, where you're working you're working the first letters in the groupings, the outside letters in the groupings, the inside letters in the groupings, and the last letters in the groupings. Okay, that's one way to look at it. Or you can say, hey, I got a capital D here, I'm going to start with that, pair it with a capital T. I'm going to keep that capital D with the lowercase t this time. I'm going to get that lowercase t this time, pair it with a capital T, and then finally the 2 lowercase letters will be paired up. Now this will overall give you kind of the outline here, and then you just fill them in. So you have B, D, T, T, all the way through, and you should get an outcome that looks something like this. Now, I went ahead and kind of color coded these so you can see what the overall phenotypes would be. So all of these overall would match up, and they would all express the same way, even though their genotypes look different. And they would give us tall and dark plants, because all these have the trait for dark leaves, and all of them have the trait for being a tall plant. That's 9 out of 16, okay? If I have a dark leaf plant that's short, that's gonna be 3 out of 16, and those would be the boxes that would correspond. You can also have light colored leaves that are tall, so I put those in yellow here, representing those, and finally in gray, this would be the light colored leaf that is short overall as a plant. Now, this creates a nice little ratio here of 9 that have the 2 dominant traits, 3 that have 1 of the dominants and 1 of the recessive traits, 3 that have 1 of the recessive and the other dominant trait, and then 1 that is both recessive. So this 9 to 3 to 3 to 1 ratio comes up a lot, and it's one of those kind of magic numbers that you should just memorize overall, because it will come up on a test or quiz at some point. When in doubt, and you see this as an answer choice of 9, 3, 3, 1, just pick that and move on. It saves you a lot of time. All right, sex link. So whenever you're talking about sex link, you have to correspond with male or female. So again, this is using that law of segregation where we split up the alleles. Now females have 2 X alleles because that's kind of what their genotype is XX. Males have an X and a Y. Okay, so what's the probability of becoming a male? Well, if you break them all down, mom can only provide an X, so every offspring has an X to it. However, the dad, the sperm cell, could either have an X in it or a Y. So in half the cases, it would carry the X, so then the match up with mom's X and make a female. Other half of the cases, it would match up with mom's X and make a male. So it's 50-50 for male or female probability. Now, why is that important? Is because some traits are actually linked to the X chromosome. So these are called X-linked traits. They're typically recessive, and they're usually on the X chromosome because there's so much more DNA on the X chromosome than the Y chromosome. So colorblindness is an X-linked trait. If a mom is a carrier, reproduces with a father who is not colorblind, what is the probability of them having a colorblind offspring? So let's look at that one real quick. Remember, the word carrier means that they hold it, but they don't show it. So mom holds the trait for colorblindness, but she is not colorblind herself. So mom is going to be 2 X's, dad is going to be X and Y. Mom carries it, so she's going to have the trait, but not show it. So I'm going to represent the trait for colorblindness as this lowercase r because it's recessive. And I picked the letter R because it's easy to tell between capital and lowercase. Okay? And then she has the dominant trait showing she's a carrier. So she's heterozygous here. Dad is not colorblind, so that means he has the dominant allele here and no recessive. Y doesn't get anything because this is an X-linked trait. So now you put the X's in there with their corresponding R's. And you would kind of see that you have a female that would not be colorblind, another female that would not be colorblind, a male that would not be colorblind, and only one opportunity for a colorblind offspring here. So if they have a offspring, it's a 25% chance that offspring is colorblind, and that offspring would have to be male. There's no outcome for a female to be colorblind here. So rounding out our last topic within this unit was pedigrees. Now pedigrees, remember, are kind of a diagram, like a family tree that shows the inheritance of a particular trait. So it's just showing that movement of the trait throughout the generations. The trait could be recessive, it could be dominant, it could be sex-linked, it could be what's called autosomal. So you just kind of have to look for some patterns to figure out what is going on overall. Okay, remember that squares represent males, circles represent females. Any horizontal lines represent a marriage or some sort of reproductive event here. And any vertical lines coming out would be the overall offspring. So you can see that this couple here had a son, a daughter, and another daughter, okay? And so this individual here marries in because notice there's no relationship above, so they're a married in individual, just like this individual married in as well, okay? And then you can see the offspring throughout the generations. The 1, 2, and 3 here on the side in Roman numerals represent which generation it is, and then the numbers underneath each individual represents their birth order. Sometimes they restart them with every generation, sometimes they keep it as one continuous flow. It doesn't really matter either way, just kind of pay attention to what they're doing on each chart. Now, let's look at this one overall to kind of figure out the genotypes of some individuals. So if I have individual 2, 5, so individual 2, right, representing generation 2, individual number 5. This person right here, what is their genotype? Well, the first question we have to ask, is this a sex-linked trait or an autosomal trait? The easiest way to tell if it's sex-linked is to see the number of males that are affected. If more males are affected, it is most likely sex-linked because they only have 1 X chromosome, so the likelihood of them showing a trait is much higher than females who have 2 X chromosomes and could be carriers or have a trait that could cover it up. Right? So, with that being said, I notice on here I actually have more females representing the trait than males. So that is a common sign for me to say that it's an autosomal or just a regular body trait like hair color or eye color or something like that. So this individual here, because it's more of an autosomal trait and it looks like it's recessive, and the reason I can tell that is because not a lot of people have it. Dominant traits would obviously show more. So this person right here probably has 2 lowercase letters. I'm going to pick the letter A because again, easy to tell upper or lowercase. And the letter A here kind of represents 2 recessive alleles, so a homozygous recessive for this individual. Let's look at individual 3-9. So generation 3 coming over here to individual number 9. So this one doesn't show, obviously, the trait, so they have to have a dominant. But the question is do they have a recessive? So they do have a dominant trait because, again, they're not filled in. Now we look back to see their parents, okay? Well, obviously, dad here probably gave a dominant trait because he's not showing any color. And then mom, though, is recessive, right? She is homozygous recessive because she's showing the trait, whatever it is. So obviously, she only has lowercase a's, so she must give a lowercase a, which means that individual number 9 here would be represented as a carrier or a heterozygous genotype. Just to show you an example of one that is X-linked, again, the key is to look for is there more males with it than females. So if I look at here, I have 1, 2, 3, 4, 5, 6, 7, 8 people that are totally affected here. I have 1, 2, 3, 4, 5 that are male and 3 that are female. So that is my sign to say, okay, it's more males than females, even though not a huge number more, but still it's more, 5 greater than 3. more likely that is going to be an X-linked trait, and this one it even helps us by telling us, okay? So I would anticipate individuals like this, like individual 15, his genotype should be an X with a lowercase letter and a Y because he's showing that recessive trait. Individual number 8 up here, she's going to be 2 X's because she's female, and she's going to have lowercase r's to represent her genotype as well. This one, this male here, would be an X with an uppercase R because he does not show it. And then, the tricky one, this individual here is most likely X with an uppercase R because she doesn't show it, and X with a lowercase r. The reason I can say with her with confidence is a carrier is because her mom shows the trait. If you look up here, you can see that mom actually has the trait and expresses it. So mom's genotype would look just like this one over here, the 2 X's with the 2 lowercase r. So mom can only give that lowercase r, therefore it's going to be a heterozygous or a carrier for this individual here. Okay? Hopefully that helps you understand a little bit more about Unit 6 and prepare yourself for any tests or quizzes you have coming up. That's all I got for today, and have a good one.